Good morning. This is Dr. Rajesh Yadav, Assistant Professor and Head in the Dialysis Technology Program, School of Allied Health Science, Sharda University, Greater Noida. The topic is erythropoiesis. Erythropoiesis means formation of the RBC. So we will see here mechanism of erythropoiesis, maturation factor which is required for the erythropoiesis, vitamins required for erythropoiesis, and hormonal control of erythropoiesis. So first come to the introduction of the erythropoiesis. Erythropoiesis occurs within the 3 months in fetus yolk cells, while between the 4 to 6 months it occurs in the liver, and erythropoiesis occurs in the bone marrow when 6 to 9 months. So increased level of physical activity can cause an increase in the erythropoiesis. In human with certain disease and in some animal, erythropoiesis occur outside of the bone marrow within the spleen or liver. This is termed as extramedullary erythropoiesis. So we have seen that erythropoiesis occur in 3 trimester. In first trimester in fetus and yolk cells, while in second trimester in the liver, and in the third trimester in the bone marrow. So we'll discuss about the maturation factor which is required for the erythropoiesis, like hypoxia. It will increase the erythropoietin hormone that will responsible for the synthesis of the RBC. Growth inducers, vitamins. Vitamin include vitamin B12, folic acid, vitamin C. Some proteins which are required for the globin synthesis, and some minerals like iron and copper which is required for the heme synthesis. Now this is the pathway or mechanism of erythropoiesis. So erythropoiesis occurs from the pluripotent hematopoietic stem cells. It will differentiate and form colony-forming unit spleen, that further differentiated colony-forming unit blast, and further differentiation and form colony-forming unit erythrocyte, which form the erythrocyte or RBC. In the second pathway, pluripotent hematopoietic stem cells form colony forming unit spleen, and it differentiates into colony forming unit GM, that means granulocyte monocyte. Granulocyte has of 3 types: basophil, acidophil, and neutrophil. So it will differentiate into the granulocytes and monocyte. Monocyte form from the macrocyte cells, while there's another pathway, colony forming unit spleen differentiate into colony forming unit megakaryocyte. So megakaryocytes, it form the platelets, while the another pathway, pluripotent hematopoietic stem cells, when differentiate into the another cells, it also make the self-copying, and through the another pathway it form lymphatic stem cells which further synthesize T lymphocyte and B lymphocyte. In this way, all the blood cells are synthesized through the process. Now this is the pathway of erythropoiesis. We have already discussed that pluripotent hematopoietic stem cells that will form the RBC. So here some interleukins are involved that will differentiate into colony forming unit blast and colony forming unit erythrocyte. It is further form proerythroblast, which form basophil erythroblast, and it will differentiate into polychromatophilic erythroblast that will synthesize orthochromatic erythroblast, and then reticulocyte which further form the RBC. So these are immature cells which are synthesized in the pathway of RBC, and most popular reticulocyte cell is also called immature RBC because it has nucleus, while when it differentiates into erythrocyte, the nucleus is disappeared. So in the human RBC, nucleus is not present. The mammals, RBC is not present in the nucleus is not present in the RBC, exception camel and llama, which have nucleated RBC. Now come to the regulation of erythropoiesis. Research study have shown that peptide hormone, that is hepcidin, may play a vital role in the regulation of hemoglobin production and thus affect erythropoiesis. The liver produces hepcidin, and hepcidin control iron absorption in the GIT or gastrointestinal tract and iron release from the reticuloendothelial tissue. Iron must be released from macrophage in the bone marrow, which is incorporated in the heme group of the hemoglobin in erythropoiesis. So we know that hemoglobin is the main component of the RBC, and it is also a pigment that provides the color of RBC, red color of RBC. So erythropoiesis is controlled by the hormone erythropoietin, which is secreted by the kidney in response to hypoxia. It stimulates red blood cells in the red bone marrow or yellow bone marrow. Common cause of cellular hypoxia resulting in elevated level of EPO. EPO means erythropoietin up to 10,000 milliunit per ml. Include anemia, hypoxemia due to chronic lung disease. Erythropoietin is produced by the interstitial fibroblast in the kidney. The association of peritubular, capillary, and proximal collimated tubules. Now these are artificial erythropoietin hormones. It is called epoietin alpha and epoietin beta. Recombinant human erythropoietin is produced by the recombinant DNA technology in cell culture and are collectively called erythropoiesis stimulating agent. So example of erythropoiesis stimulating agents is epoietin alpha and epoietin beta. So the mechanism of action of erythropoietin. Erythropoietin has been shown to exert its effect by binding to the erythropoietin receptor. EPO binds to the erythropoietin receptor on the red blood cell's progenitor surface and activates JAK2 signaling cascade. This initiates STAT5, PIK3 and RAS-MAP kinase pathway that result in the differentiation, survival and proliferation of the erythrocyte cells. SOCS1, SOCS3 and CIS are also expressed, which act as negative regulator of cytokine signal. High level of erythropoietin receptor expressed is localized to erythropoid progenitor cells. EPO receptor are also found in the heart, muscles, kidney and peripheral central nervous tissue. Indirect dependence of red cell longevity in the blood on plasma erythropoietin level has been reported, a process termed as neocytosis. In addition, there is a conclusive evidence that EPO receptor expression is upregulated in the brain injury. EPO is synthesized mainly by the interstitial cells in the peritubular capillary bed of the renal cortex, with additional amount being produced in the liver and pericytes in the brain. Parasite is also called rugged cells. It is multifunctional neural cells, the microcirculation that wrap around the endothelial cells that line the capillaries and venule throughout the body. During the normoxia, GATA2 inhibit the promoter region of the erythropoietin. GATA2 level decrease during hypoxia and allow the promotion of erythropoietin production. Thank you.